Welcome to this second video on bacterial genetics. In this video, I'm going to focus on bacterial mutations. Now it's easy to study genetic mutations in bacteria because they are inexpensive to grow. And they also grow relatively fast compared to other organisms. Plus, they only have one chromosome. Therefore, we only have to worry about making a mutation in a single gene and not worry about multiple alleles or copies of the same gene that you'd see in diploid cells. Now, because they grow fast, we can see the results of these mutations within days or hours even. So in this video, I'm going to talk about the various types of mutations that tend to occur in bacteria, as well as the classes of agents that cause these mutations to occur. And finally, I'll mention the 3 different ways that bacteria can spread their genetic material to each other. So before I go into details on what we know about bacterial mutations and how we can use that information, let me give you a few definitions. First of all, let's define what a mutation is. Now, a mutation is simply a heritable change in the nucleotide sequence of DNA. In other words, if the DNA sequence has changed for any reason, it is now a mutation. Any organism that has a mutation in its DNA is now referred to as a mutant. And the bacteria that has what is defined as the original DNA is referred to as the wild type. Now, this wild type organism is chosen based on the fact that it is the one that expresses the most common phenotype. So how do we know if a bacterial cell is a mutant or not? That can actually be kind of tricky, but we have 2 ways of finding out. The first is by looking to see if the bacteria has unusual or different characteristics than what is considered normal. These characteristics are defined as the cell's phenotype. Maybe the bacteria grows under different conditions, or it has a different color, or it might just require different nutrients to grow, etc. So these are just few examples of different phenotypes. Phenotypes are something that we can see visibly and are usually based on the protein level. Now, some mutations may not lead to a different phenotype than that is expressed normally by the wild-type bacteria. So the only way that we can know the difference between these mutants and the wild-type organism is to compare their DNA, so to look at the DNA level, not the protein level. With today's technology, it allows us to isolate and identify chromosomes' nucleotide sequence. If their DNA differ in any given way, then we say the 2 organisms have different genotypes. A genotype is basically a collection of genes each organism contains. Now, in this case, the difference between a mutant and a wild-type organism is not based on their phenotype difference, but on the difference of their genotype, or their DNA. As you can imagine, there are thousands of possible types of mutations that can occur every day in nature. So in this section, we will only look at a few examples to give you an idea of how these mutations may or may not affect how the organisms live. So let's begin with the simplest type of mutation. These are called point mutations. Now, a point mutation is when a single nucleotide has been replaced by another. So you take one out and you add another one in its place. Now, for example, here's a wild-type DNA sequence. What if a mistake was made during this DNA replication and the thymine was actually replaced by a guanine nucleotide? This is a point mutation. In effect, the thymine was simply replaced by this guanine, and based on this information, we can't tell if the mutation affected the phenotype, but definitely the genotype is different because the nucleotide sequence is now different by 1 nucleotide. Next, the other type of mutations are called insertion mutations. In this case, 1 or more nucleotides are added to the wild-type strain without replacing any. So in the long run, you end up with extra nucleotides. So for this example, we compared this wild-type DNA sequence to this mutated DNA sequence. What you'll notice is that there's an extra G, or an extra guanine nucleotide, that was added to the mutant sequence. Now this example only shows the insertion of one nucleotide, but an insertional mutation can lead to the addition of more than one nucleotide. You can have 2, 3, 4, as many as 100 nucleotides added. In this case, if the mutated DNA has an increase in nucleotides, then it's an insertion mutation. The opposite of an insertion mutation would be a deletion mutation. This is where one or more nucleotides are actually removed from the wild-type DNA sequence as shown here. So in the long run, the DNA strand or sequence is shorter. Now the examples just shown only shows a single DNA sequence. But remember that all chromosomes are double-stranded. So keep in mind that whether it's a point mutation or an insertion or deletional mutation, the mutation actually affects both strands in the same way. So within the following slides of this video, you're going to actually see double-stranded DNA, and I'm going to compare how a single mutation or an insertional mutation will affect both strands. So I haven't yet asked or answered the question is, how do these mutations affect the organism? In order to answer this question, we first need to understand how the genetic code works. Here's an example of a genetic code shown on the right. Now remember in the first video, I showed you how ribosomes come together with the messenger RNA. They're going to look for the first codon AUG, and they're going to start interacting with transfer RNAs, which are going to bring in amino acids. So when a ribosome reads this genetic code found within the messenger RNA, the ribosome actually travels along a single-stranded messenger RNA, starting at its 5' end and ending at its 3' end. What it's doing is it's reading the messenger RNA in the 5' to 3' sequence to determine how it's going to join a series of a series of amino acids together in a unique order that will determine which protein will be made. So it's very important that the ribosomes and the messenger RNA come together specifically with transfer RNAs to make the specific proteins. Now this genetic code is a template that will tell the ribosome which amino acid it will use to add to that growing chain of amino acids. And by the way, this genetic code is universal. Basically every single organism follows this genetic code. So let's use this short piece of DNA as an example. If I act as a RNA polymerase and transcribe it into a messenger RNA, this is what the messenger RNA would look like. Notice that the DNA and the RNA strands are anti-parallel to each other. Now when they're done being formed, they are separate from each other, so they're not hydrogen bonded to each other. In this case, also the messenger RNA, instead of having thymines, it's replaced with uracils, with U's. Now the ribosome can will attach to the 5' end of this messenger RNA, and it will search for the first AUG nucleotides closest to the 5' end. And now in this example, I happen to put them as the first 3 nucleotides, but that's not always the case. Now the ribosomes will add a methionine at this first AUG. So every single protein will begin with the methionine amino acid. This first AUG is going to determine how all the other nucleotides will be grouped together in groups of 3s, also known as codons. So the ribosomes will continue sampling the messenger RNAs in these groups of 3s. Until it reaches a stop codon, where the ribosome will release the messenger RNA and the new protein is now released. So notice that this stop codon in this example is a UAG. So let's backtrack and make sure that you understand how the ribosome knows how to covalently link methionine to alanine and then to valine. So if you look at this example, you're going to read AUG, and in the genetic code, the first position are in rows, the second position are in columns. So A is the first position, U is the second, they come together to this box, and you look for the third nucleotide AUG. So it says start and methionine. You will always put a methionine. The second codon is GCG. So we go to the first position, G. C, we come down here, GCG. It encodes for the amino acid ALA, or alanine. The next is GUC, so GUC, that's valine. And then lastly, the stop codon, UAG. There are 3 stop codons. So where there's UAG, we don't write stop, we don't even write the amino acid because there are no amino acids. So this protein is made up of 3 amino acids. Okay, so as mentioned, these point mutations may or may not affect the organism's phenotype. In other words, it may or may not affect the function or even the production of the protein that is encoded by that mutated gene. So remember that a point mutation occurs while the chromosomes are being duplicated. So the point mutations occur at the DNA level. Now, if these random mutations occur within a gene, then their effect is carried over to the messenger RNA during transcription. The position of the point mutation within a codon will determine if the codon change will encode for the same or a different amino acid, or whether a stop codon is inserted. So if I go back to a slide, see if I can just, if you look here, if we have a mutation in the third position of this codon, any mutation in the DNA that would lead to a difference in the third position of the messenger RNA, we will always have the amino acid leucine. So any mutation would not be picked up at the protein level. It would not change the protein any. However, if I had a mutation in the first position, here's a CUU. If I change the CUU to an AUU, so I change a C to an A, I went from a leucine to an isoleucine, a different amino acid. So you can pretty much see that the position of where the mutation lies within the codon will have either a small impact or no impact at all. So either way, the resulting codon sequence is read by the ribosomes during translation. So here's an example where a point mutation had no effect at all on the resulting protein. Let's first look at the wild-type messenger RNA strand, and notice that the AUG is not the first nucleotide listed at the 5' end. Usually AUG is not the first group of nucleotides. However, the ribosomes always start at this 5' end and look at this first AUG. This will now tell the ribosomes, again, as I mentioned earlier, where to start reading the messenger RNA in groups of 3 as codons. Now let's change an alanine. I've highlighted this alanine right here. Now, like the wild-type strand, the ribosome starts reading the codons at the AUG start codon. If we focus at the third codon that we've changed from GUA to GUU, both of these codons tell the ribosome to add a valine amino acid at this position. Thus, this point mutation is what we call a silent mutation, because the mutation didn't affect the amino acid sequence at all. Again, how do we know that this is a mutation? We would have to look at the DNA. Now in this second example, the mutation that changed a cytosine right here into an alanine actually did change the codon, resulting in the change in the fourth amino acid. We went from a proline to a threonine. Now this point mutation is not a silence mutation because it actually changed one amino acid to another amino acid. So we're going to call this point mutation a missense mutation. A missense mutation changes one amino acid to another amino acid. In this third example, shows you that sometimes a point mutation might actually change a codon to another codon that it actually encodes from a stop to a stop codon. So in this example, I have UAC. This UAC codon encodes for the amino acid tyrosine. When we change the cytosine to a guanine, the codon now is converted to a stop codon. So we went from a codon that encodes for an amino acid to one a stop codon. This type of point mutation is called a nonsense mutation. Now what's not shown here is that the opposite could actually occur. That where you have a stop codon, that a point mutation can change that into a codon that encodes to an amino acid. That is not called a nonsense mutation. And actually I can't find the term if there is a name for that type of mutation. But when you go from a codon that encodes for an amino acid to one that's a stop codon, that alone is a nonsense codon. Now you can imagine that if you all of a sudden insert a stop codon, you're actually shortening the amino acid sequence. Likewise, if you remove a stop codon, you're going to increase the protein. So either way, you're going to modify that protein structure and you're going to affect the phenotype. Now besides point mutations, there are other mutations that instead of substituting one nucleotide for another, they actually add or remove. So I've mentioned insertion and deletion mutations. The key is whether the additional or the removal of nucleotides are in multiples of 3. Now the example here involves the removal of only 2 nucleotides, so 2 is not a multiple of 3. In this case, we've removed the uracil and the adenine, the U and the A. Notice that the AUG and the AGC codons here on the left of the deletion were not affected. So in both cases we have a methionine and a serine in the wild type and in the mutant strain. However, every codon to the right of that change, of that deletion, has now changed. So by removing 2 nucleotides, we've now caused this G to group with the C's in the codon next to it. So now the GCC becomes its own codon, a different codon, and all the other codons after that have been changed. What results does that lead? We basically have a different sequence of amino acids. In this case, we would say that a frame shift has occurred. A frame shift, or a reading frame, is the frame in which words are made, and these words are based on 3 nucleotides. So the words here would be serine, valine, proline, tyrosine. I forgot to mention methionine. Now because we changed on how these groupings occur, we have a different sentence. Now we have methionine, serine, the same, but we've lost valine, and we now have alanine, leucine, leucine. We don't have proline and tyrosine anymore. And where this was once a stop codon, that stop codon has been removed. So the groupings have changed. They've slid over by 1 nucleotide. The reading frame has been changed. So just to recap what I've just covered were the 2 major different types of mutations, the point mutations and the insertion and deletion mutations. I've also broke down the point mutations into the silence mutations, the missense mutations, and the nonsense mutations. So let's switch gears for a moment and talk about what causes these mutations to occur. So first of all, it's important that you know that these mutations are actually a part of the organism's natural lifespan. So as an organism grows and multiplies, random mutations will occur at a rate of approximately 1 nucleotide in every 1 billion that are added during DNA replication. The key word is these mutations occur during DNA replication. Now these random mutations, or these random events, will help an organism evolve or adapt to its environmental changes. However, there are some forms of radiation or chemicals that we come in contact with every day, that will help increase this rate of mutation. So instead of every 1 in a billion, it might be every 1 in a million, as an example. Now, these substances that cause mutations are called mutagens. Now, I'll warn you to try not to confuse mutagen with that of mutation and a mutant, because each of these terms have very specific meanings. So let's talk about these different mutagens. Now, there'll be radiation and chemical mutagens. So I'm going to first mention the 3 different types of chemical mutagens. The first one listed here is a nucleoside analog. Now, these are molecules that are similar enough to the DNA nucleosides that they're easily added to a growing strand during DNA replication. But at the same time, they're different enough to cause them to pair with the wrong complementary nucleoside on the opposite strand. So here's an example of how that could happen. In this example, the nucleoside analog is 2-aminopurine nucleoside, or 2AP. Now, 2AP is the analog of the nucleoside adenine. Now, remember that adenines pair up with thymines, A's to T's, on complementary strands. But in this case, the 2AP will pair up with cytosines, C's, not with T's. So what effect will that have? Now, the effects of 2AP is felt when the new strand acts as a template to make a complementary strand. So let's start with a wild-type double-stranded DNA. And during DNA replication, these 2 strands have to separate. Now, in turn, these 2 strands act as a template to make additional copies of the complementary strands, as shown here in yellow. Now, if the 2AP is taken up by the cell, some will randomly insert themselves in the new strand as opposed to taking the adenines. So at this point, the nucleoside analog hasn't really had an effect on the cell yet. We go through a second round of DNA replication. Now the cell's DNA undergoes the second round. This time, one strand has this 2AP in it. This is the strand that we need to watch out for. Again, the double strands will separate, but the strand that has this 2AP will result in a mispairing, as shown here. So remember that adenines pair with thymines, T's, but in this case, this 2AP pairs with a cytosine. So only the cells that contain this mispair will have the mutation. Now, can you guess what kind of mutation this is? Is it a point mutation, or is it an insertion or a deletional mutation? It's a point mutation. Now, all the other cells that have the DNA from the other 3 strands will be considered wild type. So it's really that 1 out of 4 events that will carry this mutation over. So when this DNA strand with the mispair undergoes a 3rd round of DNA replication, what was once an AT nucleotide pair has now switched to a GC pair. And then this is carried on from one generation to the next. Now, the second class of chemical mutagens are the nucleoside altering chemicals. Their names are very similar to that of the nucleoside analogs, but their effects are quite different, so try not to confuse the two. Now, in this case, these chemicals actually alter or change the nucleosides that are already present inside the DNA strands. Their overall effect is similar to that of the nucleoside analogs, such that the net effect is the change in the nucleoside pairing along complementary strands. So let's take a look at nitrous acid as an example. Now, nitrous acid acts on the nucleoside cytosine, and what it does is it changes its nitrogenous base so that this cytosine now looks more similar to that of uracil. Remember that cytosine pairs with guanine, and that uracil, which is usually found in RNA, not DNA, will pair with adenine. So you can imagine the effects will change the nucleosides that basically take this position. So again, here's a double-stranded DNA sequence. If we expose this double strand to nitrous oxide, one of the cytosines is modified and looks like uracil. Now notice we didn't have to go through a DNA replication here because these chemicals modify the nucleotides or nucleosides that are already present in the DNA. So we went from a cytosine to a uracil. Now the DNA needs to replicate. The two strands will serve as templates to make their complementary strands. And notice that the top strand has a uracil mutation, whereas the bottom strand remains a wild-type strand. After the complementary strands are made, the upper double-stranded DNA now has a point mutation, and depending on where it's in the strand will determine if the mutation will have an effect on the protein. So in effect, we went from a GC to an AT mutation, a point mutation. The third and last class of chemical mutagens are the intercalating agents. Now these are molecules that insert themselves in between two complementary strands of double-stranded DNA. So we call them intercalating because they intercalate or insert between two strands of DNA. By doing this, they're going to create a stress on the strands and form a bulge in the helix. Now the cell's DNA polymerase are going to see this as being unusual and will want to fix it, as shown here in this figure. Now in doing so, half the time the repairs will lead to a deletion of nucleosides, as shown here in the bottom left corner. While the other half of the time, the repair will accidentally add more nucleotides, as shown in the bottom right corner. So again, the intercalating agents, in this case acridine, inserts itself between two strands of DNA. The DNA polymerase will come and remove and try to repair that sequence. Half the time it will cause a deletion mutation, other times it will cause an insertion mutation. And in both cases, it could lead to a frame shift mutation. So that's it for the chemical mutagens. Let's look at the two radiating mutagens. Let's start with those of the ionizing. Now the ionizing radiations include X-rays and gamma rays. Both of these radiations will penetrate the cell's nucleus and damage the double-stranded DNA by breaking the sugar ribose backbone, as shown here. Now, as the cell's DNA polymerase attempts to repair the breaks, the affected nucleosides are removed and replaced. But again, errors are sometimes made, 1 in every billion. So we're increasing the number of nucleotides that are added. So we're increasing that rate of mutations. Now, the second form of radiation mutations, the non-ionizing radiation. In this case, the example are UV or ultraviolet light. And on this hand, they will affect the DNA where there's only two thymine nucleosides that are adjacent or next to each other on the same strand. Now, normally, these nucleosides are not covalently linked together by their nitrogenous base. However, by exposing the DNA to this UV light, the thymine nucleoside nitrogenous bases will actually form a dimer. They will covalently bind to each other. Now the two thymine nucleosides nitrogenous bases will form this bulge in the double strand. Very similar to the intercalating agents, except in this case it's only one of the two strands that form a bulge. Now in this case there's two ways of repairing this bulge, or to separate the thymines. Now in the first case shown on the left, the cell's nuclease enzyme will excise or remove part of that double-stranded DNA, and then the DNA polymerase 1 will come over and backfill. It will add the correct nucleosides. This is shown here in the left side of this figure. But sometimes the repairs are made by a photoreactivation pathway, so it needs light to activate this pathway. This is shown here on the right. So in this case, the enzyme photolyase, in the presence of light, can simply break the covalent bonds that bind the two thymines together. So that basically summarizes the 5 different types of mutagens, 3 that are chemical mutagens and 2 that are radiating mutagens. So you should be able to give me an example and understand how these mutagens affect DNA. Now, scientists are constantly looking out for potential mutagens. Any chemical or radiation that we come across during our lifetime should be suspected as being potential mutagens. Therefore, companies of all industry types need to test their products before they're actually sold for human use. To better understand how this is done, let's first look at a few definitions. So the first one is replica plating. This is a widely used technique that helps a microbiologist identify bacteria that have a unique nutritional need, meaning that these bacteria will only grow if this need is met. These bacteria are called auxotrophs. So for an example of an auxotroph is a bacteria that cannot make the amino acid histidine. Now I'm showing you the pathway here in the lower right hand corner of the slide. In order for a bacteria to not be able to make this amino acid histidine, it needs to miss one of these enzymes, or one of these enzymes have to have a mutation that prevents it from functioning. In this case, this bacteria is called a histidine auxotroph. Now, a bacteria that can make the amino acid histidine doesn't need to have it added in its media in order to grow. Now this bacteria, instead of being called an auxotroph, it will be called a histidine prototroph. This bacteria will grow whether or not the amino acid is added in its environment. Thus, replica plating will allow a microbiologist to identify the auxotrophs from the prototrophs. And the key point here is to actually identify the auxotrophs. So here's a setup of replica plating. We always start with a culture of bacteria that are grown in the presence of histidine. Remember, both auxotrophs and prototrophs can grow in media that have histidine. So in this way, if a mutation has disrupted a function of any of the genes that are required to make histidine, then that organism, an auxotroph, will still grow. Remember, that's the one that we want. So in effect, this culture should contain the mixture of auxotrophs and prototrophs. So now this mixed culture is plated on an agar plate that contains media that has histidine in it, as shown here in step 1. These colonies are allowed to grow, and then next, a sterile felt cloth is gently pressed against these colonies. Now this cloth is used as a stamper to transfer the pattern of colonies to 2 plates. As shown here in number 2, one plate contains histidine and the other plate does not. Now the ability to replicate the exact position of each colony is key to identifying which organism is the auxotroph. Now these 2 plates in step 2 are then incubated to allow the bacterial colonies to grow. And at that point, the patterns on both plates will be observed, and they should be the same pattern as that as shown in step 1. Now if a colony is shown to grow on the plate that has histidine, but does not grow on the plate that lacks histidine, as shown here in this red circle in step 4, we are going to label that organism as an auxotroph. Now imagine if we had only plated these organisms on the plates lacking histidine, the auxotrophs would not grow, we would never see them. So we actually need the plate that contains histidine to show that it is present in the culture. Now all the other colonies on the plates that grow in both plates are the histidine prototrophs. So this experiment is very important to collect a number of auxotrophs for the next step that's needed to identify potential mutagenic compounds. Now this is the same slide showing the histidine pathways I'd shown you in the lower right hand corner of the previous slide. It shows that of the pentose phosphate metabolic pathway. Now this pathway is responsible for every cell to have the ability to make histidine. Now I'm showing you this because I'd like you to notice that there's at least 9 genes that are labeled HIS, H-I-S. This is short for histidine. These 9 genes are needed to fully they need to fully function in order for the cell to make the amino acid histidine. And again, it only takes one mutation in any of these enzymes to convert the bacteria from a histidine prototroph into a histidine auxotroph. Now this table lists 5 different Salmonella typhimurium bacteria that are known to have unique mutations in either the histidines or HISD, G, or C genes. These are just examples of auxotrophs that were identified to have either a point mutation, in this case they're labeled as base pair substitutions, or to have a frameshift mutation. Now these auxotrophs are going to be used collectively, meaning all together, in the next test that's known as the Ames test. Now this Ames test is a rapid, very inexpensive method that will use histidine auxotrophs. So remember, the replica plating allowed us to identify the auxotrophs. The Ames test uses these auxotrophs. These histidine auxotrophs are going to be used to screen for substances that may or may not have the ability to mutate DNA. Now the theory is if they can mutate DNA, then these organisms have the potential to be also not just mutagenic, but carcinogenic, meaning that these mutations can lead to cancer. Now the theory that basically is the basis of this Ames test is that if exposure to any given substance leads to an increase in mutation rates, then the exposure of this substance will also lead to a greater cancer risk. Now remember, we're going to measure this increase in mutation rates by the conversion of histidine auxotrophs into histidine prototrophs. And the bacteria that we tend to use a lot during the Ames test are the Salmonella typhimurium bacteria that are histidine auxotrophs. So in the Ames test, what we want to detect is, will these auxotrophs convert to prototrophs? And if so, if it's substantial, then this organism, this substance is a potential mutagen. Okay, so let me walk you through the Ames test. In this case, you always have 2 test tubes. This one is the background test reaction, or control tube, I should say. And this is actually the tube that tests for the reagent that is a possible mutagen. Now in both of these tubes, we're going to have Salmonella typhimurium histidine auxotrophs that are present. So we're going to have histidine in the media so that these auxotrophs can grow. We're going to add rat liver extract to both of these tubes. Now rat liver extract is because when chemicals tend to filter through the livers of our bodies, if the chemical is not mutagenic, then it might be after it goes to the liver. So there's some reactions within the liver that can convert substances into potential mutagens. So we're going to add these rat liver extracts to both tubes in order to increase the chances that if these chemicals are mutagenic, that we will see it. In the test reaction, we're going to add our potential mutagen. In this case, it's this blue colored solution. Notice that that is not present in the control tube. Now, after a time of exposing these cultures, in this case to this potential mutagen, both of these are going to be plated on plates that do not contain histidine. So these bacteria are plated on media lacking histidine, and the number of mutants that regain the ability to synthesize histidine will be recorded by counting the number of colonies on the plate. Now remember, it takes only one enzyme to lose the ability to make histidine, but we have to actually convert that histidine back. So we have to revert that mutation that occurred in that one enzyme so that that one enzyme can gain its function back to go from an auxotroph to a prototroph. Now you notice in this tube here on the left that we have 2 colonies. Now these 2 colonies are due to spontaneous mutations in those enzymes that were mutated. So they spontaneously reverted back to prototrophs. So this is our basal level. This is our background noise, so to speak. On this case, by the simple fact of adding this potential mutagenic chemical to the bacteria, we actually went from 2 colonies to, I don't know, maybe 20 colonies. So the number of histidine prototrophs has increased substantially here. So by this reaction, we could say that this possible mutagen is a mutagenic chemical. What I should also mention is, based on the number of prototrophs that appear on this plate, we can then determine how mutagenic this chemical is. So now that this Ames test has been performed and we've identified this chemical as a potential mutagen or as a mutagen, this compound has to now go being tested in animals or animal models to then determine whether it is carcinogenic. So again, just the simple fact that this chemical is mutagenic doesn't necessarily mean that it is carcinogenic. At least in this test, we did not prove that step. So we now need to take that mutagen, expose it to animals to see if it causes cancer in the animal model. Now what's not shown here is just for the fact that if this possible mutagen was not a mutagen, we would basically just see 2 colonies. So just to give you an idea that not all chemicals are mutagenic. If they are not, you would see basically the same number of colonies on both plates if they were non-mutagenic. Okay, so that's going to end the section on mutagens. We're next going to move on to how bacteria achieve genetic diversity. Now, using bacteria as a genetic model has its benefits as well as its limitations. So, for example, organisms that are capable of sexual reproduction can achieve genetic diversity during the crossing over events that are seen in prophase 1 of meiosis, as well as during random fertilization during the fusion of their 2 gametes. But because bacteria only have 1 chromosome, they are haploid, and for this reason, they don't have to undergo meiosis. Hence, they have less genetic rearrangement. Instead, also they only replicate asexually by the form of binary fission. But this doesn't mean that they don't exchange DNA and do not experience genetic diversity. In fact, bacteria can acquire DNA from other organisms by 3 different methods. These methods are transformation, transduction, and conjugation. So I'll go into these a little bit more detail. So during transformation, naked DNA is taken up from the environment. Now in this example, I'm showing you that it's a plasmid. Remember, plasmids are circular double-stranded DNA, but these pieces of DNA can also be linear. Now, some bacteria have the ability to absorb DNA that was given up by their neighbors, other bacteria, that happen to die in the vicinity and release their DNA into the environment. This DNA can be, as I mentioned, pieces of chromosomal DNA that is ultimately exchanged, or so inserts inside the chromosome of the recipient bacteria. But it can also be these small circular pieces of plasmid, which can either stay independent or they can also enter the chromosome. So in either case, DNA from one bacterial cell is then taken up into another host cell, another bacterial cell, and they can exchange DNA in that way. As for transduction, they're going to use the vehicle of bacteriophage. So bacteriophages are viruses that infect bacteria. We call them bacteriophage, or sometimes for short we call them just phage. Now, a bacteriophage will infect a bacterial cell. It will be released, and at times it might take up the host DNA from that bacteria, and then go on and infect a second bacteria in which it will transfer that DNA. So DNA is transferred from one bacterial cell to another through the aid of this virus particle. And the last form of transferring DNA from one bacteria to another is through the form of conjugation. Now, these bacteria have to have the ability to make a hollow tube called the conjugating pilus or the sex pilus. As it does this, it will transfer some DNA from one cell to another. Now, usually the genes that allow the cells to make this pilus are found on a plasmid, and that's the plasmid that is the plasmid DNA that's being threaded from one cell to another. It's not the chromosomal DNA. However, this plasmid will also contain other genes besides the genes needed to make the pilus. So, in this case, bacteria can transfer genes that help it be resistant to antibiotics or to be pathogenic. It could transfer one gene from one cell to another. So while eukaryotic organisms don't exchange DNA in these manners, microbiologists have used these three abilities to transfer foreign DNA into their bacteria in order to study them. So these are good tools to use in the laboratory. We can also use them in biotechnology as a way of producing human genes inside bacteria. So this is going to conclude this second section on bacterial genetics.